Hi everyone. So for week 5, we will be looking at how stress affects cell membranes. And so just to briefly summarize the utility of cell membranes, membranes serve some really important functions for cells, like determining the rate and what kinds of molecules enter and exit the cell. They also give order to the cell by separating its protoplasts, or organelles in cytoplasm, from the outside environment. And stresses like temperature and chemicals can break down membranes and reduce the effectiveness of this barrier. So for lab, we're going to study beet cells as an example of this. And beet cells contain beta-cyanin, which is pigment found in their vacuoles. And by exposing beet cells to two different chemicals, acetone and ethanol, we can observe how exposure to different concentrations of chemicals disrupts cell membranes. And so for the rest of this video, we'll review the protocol and specific steps involved for this week's lab. So the first step is to extract a beet core using the cork borer. The core will be about 5 millimeters in diameter, and it might take some pressure to push the borer through, but when it comes out, it should be a homogeneous shape. For the next step, you will soak your beet samples in tap water for 2 minutes. This is to wash the beta-cyanin from the surface of the cells that were punctured while carving out your sample using the borer. For the third step, remove your beet samples from the solution and discard your water. Then cut your beet sample cylinders so they're about 5 millimeters in length. And then next, add the beet cuts to solutions of acetone and methanol. So once you've added your beet samples to your solutions of acetone and methanol, allow them to soak in the solutions for 20 minutes. You should gently shake or agitate the solutions every so often to help digest the cell membranes. And when the 20 minutes is up, pipette 2 milliliters of each solution into a separate cuvette. The final step is to measure the absorbance of each solution. After turning on the spectrophotometer using the switch on the side, set the wavelength to 536 nanometers. Then place a blank solution in the machine to calibrate. Finally, measure and record the absorbance of your acetone and methanol solutions. It's a good idea to recalibrate the spec after every several runs.